we are ready to go with our session today. Hello, everyone, and welcome to the Graduate School One Health Emerging Infectious Diseases webinar series. I am Paola Gamboa, Education and Digital Manager of the Graduate School. Thank you for joining us for this new session in our series, One Health Research in French Amazonia, led by Professor Loic Eppelborn. We are happy to have you here with us today, and glad to know you are joining us from Philippines, Nigeria, Zimbabwe, Ethiopia, Pakistan, Israel, Ecuador, Nicaragua, Argentina, Sweden, Germany, French Guiana, and mainland French. I am pleased to introduce our chair for this session, Professor Loic Eppelborn, our expert in infectiology and epidemiology of infectious and tropical diseases. He is a full professor and hospital practitioner in the Infectious and Tropical Diseases Unit at Cayenne Hospital, Centre André Rosemont in French Guiana. Somebody, I think, is having an extra micro open. Please, could you close it? Thank you. Today, we are happy and particularly honored to welcome Jean-Bernard Duchemin. On April 22nd, we will welcome Professor Guillaume Audin. On May 18, Professor Antoine Adenich. And on June 24, Professor Paul Letournier. Remember that all our sessions take place at 12:30 p.m. Paris local time. A few quick reminders before we get started. The 40 minutes presentation will be delivered in English, but you are welcome to ask questions in either English or French. We encourage your active participation. Feel free to share your thoughts in the chat at any time during the talk or during the discussion period. When the discussion begins, you are invited to turn on your camera and introduce yourself by sharing your name, your role, if you are a student, researcher, or other, the institution you work with, and the country you are joining us from. Remember that the videos of the previous session are already available on the Graduate School website in the media section, the webinar series entry. I will share the link with you later on during the session. Well, for now, I wish you to enjoy the session. Professor Eppelborn, you have the floor.
Thank you so much, Paola. And I can see on the chat that we've got also people connected from Democratic Republic of Congo. So welcome. And I think for the first time, someone connected from Guyana, our almost neighbor at the west of the French Guyana. So welcome to all. Thank you so much to all to be connected. Thank you, Paola, for organizing this webinar. Thank you also to Professor Solène Kernis, who is helping us also, and to this very interesting project that highlights research on what's happening in French Amazonia and more generally in the Amazon and the research in the Amazon. And that's a new aspect of the research in the French Amazon today with my colleague and friend Jean-Bernard Duchemin, which is at the same time a medical doctor and also researcher, and he's been conducting some researches on mosquitoes, especially Aedes and Anopheles in French Guyana and elsewhere in the Institut Pasteur of French Guyana. So I'm very pleased today to have him to present the research on this very important topic. As you know, diseases such as malaria is still endemic in French Guyana and all the Guyana Shield. We've got a re-emerging infection of chikungunya, which is arriving from Brazil, Cuba, and Suriname to French Guyana. So this is so important. This is so actual topic. So Jean-Bernard, thank you so much to be here today, and the talk is yours.
Okay. Thanks very much, Loic, for your kind introduction. Indeed, I won't talk about chikungunya or very little or not too much about Egypti vectors. So we will talk about. I will present you some mosquitoes, on ideas about mosquitoes, about bridges on the main concept that I try to defend, that the mosquito is not a flying syringe, and then have some reflection on some ideas about the Amazonian region as an importer or exporter of virus, with concluding remarks. So, as I told you, Aedes aegypti is out of my topics, despite that beautiful mosquitoes here on the screen, because I wanted to emphasize the idea of One Health, and indeed, Aedes aegypti is really biting human and very few animals around, very linked to human and enterprise region and urban areas. So I will underline some other aspect of the mosquitoes of French Guiana. So mosquitoes, here it's a part of our collection in the lab. You see many boxes full of specimen kept as a reference for our collection and our identification skills. Here you see on the right Stanislas Talaga with the expert in mosquito taxonomy. You see that we try to go from mosquitoes that we trap in traps, we capture in traps, such as this one is on the right is Sabetes quasicyanus, magnificent mosquitoes, blue, metallic blue in the forest. And then Stan tried to decipher the mosquitoes, especially about a big focus on Culex mosquitoes, because they are very difficult to differentiate as females. So the idea is try to find tools for helping us on everyone to try to determine the Culex species we are really working on. So an example here, you see on the left, bottom left, part of the genitalia, of the male genitalia of Culex. It's so the genitalia male are indeed a sort of forceps with pairs of forceps and then a median phallosome in the media. Much more complex than what I've described you in a few words. And Stanislas is really an expert in that. So he tried to, he succeeded in linking that sort of morphologic criteria with sequencing, DNA sequencing, with part of the gene CO1, cytochrome oxidase subunit 1, which is a universal barcoding tool, which is really interesting, and that make really a good input of the lab and output of the lab, because we have now barcoding nearly 80% of the species. As you see on the text, there's more than that. Now a bit more of 250 mosquito species in French Guiana. Definitively, given the size of the territory, that's a hotspot for mosquito diversity. So just an example here of a description of a new species. So that's Culex carency in honoring our technician Romuald Carency here. Why is this one? Because for when you see in the frame on the right, it is part of the Pedroi subgroup. So it's close to Adamesi, to Pedroi, to Portesi, and we will discuss a little bit about Portesi. Almost all these vomerifera, to all these species, has been described as carrying, I don't say transmitting for the moment, but at least carrying virus, and they are really interesting suspected vectors. So mosquitoes are interesting for us because they are biting, and they are first upstream attracted before the biting process by the odor of animals, human. And so there's some field work about that. That's always a bit complex. So, for example, on the left, we have double bed net where we can put, for example, cows or calf inside. Here on the right, it was a bit old works we've done in Madagascar with two tents, one with a human, one with a calf, and we can check in the on-try cages which mosquitoes are attracted by calf odor or by human odor. Roughly same weight and same CO2 production to compare. So that's an example. So that's relatively heavy. So what we have tried to do was much more to look after the preference of mosquitoes on the downstream process. That means after the bite, once the mosquito have bitten, on the rest in the environment. So the first step is sampling. So try to find resting females. As you see here, Guillaume Lacour is aspirating, vegetation around cages in the zoo. And then we sort out for engorged females. And after we are sorting out a second time for Culex species, because that was a focus of Amandengides here on the top right, to study the Culex pattern of preference for the host. And finally, she has screened for DNA for finding the blood meal origin with several markers for that. The interesting part of that is that with NGS technique, we can try to make analysis of mixed blood meals, and I will show you a bit some data after. So here in the zoo, so why the zoo? Sorry, I forgot to mention that. The zoo is interesting because it's a relatively controlled choice. That means that the zoo is only harboring local species of vertebrates, and the mosquito have a range of hosts where they can choose the best, the ones they prefer and they fit for. So typically you see the zoo vertebrates present in cages on the histogram on the left. On the right, you have what we find in blood meal of mosquitoes. And clearly, the distribution of hosts as mammals, birds, squamate, and amphibians are roughly the same. So there's no amphibians in cages in the zoos, but clearly, mosquito could find amphibians in the environment of the zoos, and you can imagine that frogs could be found in that environment. So clearly, the distribution is roughly the same. So that proves that the mosquitoes are really good samplers of their environment, at least for vertebrates, and you see that. So an example here. So you have on the left diagram, you have the left column, which is mosquitoes of the which have been found in French Guiana. And then on this same diagram's right part, the host. So you see, for example, the Culex portici, top of the diagram. All the lines are linking to some mammals, but also to some birds. It's not easy to see on from what you know, but please trust me. There are some birds which are also bitten by Culex portici. On the bottom part, Coquilletidia venezuelensis, you will heard of this species. It's both species, so don't worry. All the mosquito for Coquilletidia, you see that all lines are red, so definitely he only is biting only mammals there. On the other diagram, on picture on the right, you see all the mixed blood meals, and you see that sometimes in the frame you can have some meals which are harboring three sources of blood meals. That means that the mosquito have bitten three different the same samples of mosquito species have bitten three different hosts in one blood meal. Or at least at the end, once the midgut is full of blood, it means it has been fulfilled by three different hosts there. So that's really something interesting. Another picture is here, so that's roughly the same idea that previously shown, but maybe a little bit clearer. So you see for the genus Coquilletidia, in blue Coquilletidia venezuelensis, mainly on indeed 90%, more than 90% are of mammals origin. And you see here that the human is not anecdotal. Merely, I think it's about 35% of blood means from Coquilletidia venezuelensis, which have been taken on human. So these samples came out from the zoo. So that's interesting because Coquilletidia is a mosquito which has a very wide range of biting out time. So it can bite during the daytime where humans are visiting the zoos. So that's interesting. And you see only one bird in that case. It's a Gallus gallus, so it's a chicken, probably from a farm nearby. On the other part, on the right side, you see for the culex, so the birds are in yellow are a little bit more, much more represented than for the Coquilletidia genus. And the human is a very small square at the bottom bottom right of the picture. And you see, why is it so few human? Indeed, culex are night biters mostly, and there's no human in the zoos during the night. So that underrepresentation of human in that case is probably a bias, because the choice, I said the choice for the mosquitoes was underrepresented for that genus because of the habit of biting out time of culex during night time. So mainly the ideas I want to share with you is that mosquito, and even one species or several species, could bite many things. You know, for culex, for example, in Europe, we used to have the idea of culex biting mainly birds, where in that case you see that there's a large range of hosts which could be bitten by. So that proves that clearly mosquito can exchange potential pathogens between sectors, between birds and mammals and human and even reptilia, etc. So that drives me towards the idea of cycles. So you have on the left the urban cycle in red, involving only human and very domestic vectors. So clearly, Aedes aegypti is one good representative of that. Aedes albopictus could be also classified as urban in urban cycle, or Culex cancer too is really linked to humans, so that could be clearly included in this urban cycle, which clearly appear as relatively simple. On the other side of the picture, you are on the right, you have the epizootic cycle in yellow. And in that case, instead of human, you have domestic animals where vectors, which are named epizootic vectors because they could trigger epizoocy in animals. Clearly, they have to like to bite animals. But sometimes you see there's a small arrow towards human because they could transfer, for example, a virus here from the epizootic cycle towards human, and that may be the idea for Japonesis on encephalitis, for example. And in the middle, in blue, is a more complex cycle, which is enzootic selvatic cycle. So instead of domestic animals, you have wild fauna, vertebrates, hosts, rodent, monkeys, birds, marsupials, etc. when it's present. So it could be a very wide and diverse fauna. And in that case, the enzootic vectors at the same could bridge towards human. That's why some of the species are called bridge vectors, because they could bite both wild fauna on the human. In that case, you have the idea of spillover. Here we will go a little bit further. So there's many. So I show you the concept of onzootic amplification in case of onzootic because of the animal farming, which could trigger a big reservoir for virus, for example, and amplify circulation. Urban cycle, you've seen that. So the notion of spillovers, it's really the notion from wild cycle, onzootic cycle towards human, where there's a sort of spill of the virus beyond the classical barriers between the forest, for example, on the villages, on the human, and you have that concept of spillover. On mirroring that concept, you have the spillback. That means that you could have a spill from the human compartment towards the wild fauna. One good example is the yellow fever in South America. You know, the yellow fever virus has been brought by slave trade in the old centuries. And then that virus with human circulation in forests has been spilled back towards wildlife in South America with sanctuaries after on autonomous circulation of this virus in South America. Here on the picture, you see many factors which could favor that either spillover or even spillback on towards domestic animals, human or wildlife, with clearly the environmental change with human encroachment towards forest deforestation and so on logging, gold mining, etc. Many factors which could favor the exchange between these different sectors. So, despite these sectors are considered autonomous and that vector could fly from one sector to another, they are not only flying syringe. So mosquitoes are living organisms. They interact with the environment, with their host. We already see that, and with microbes towards with the concept of vector competence. I will go deeper a little bit later. But what's interesting is that these interactions are really specific, and you have to keep in mind, the notion of receptor of cells, and so that's really a interaction at the epithelia, interaction in the defense rank and defense means that mosquitoes have, etc. So you have an evolution between the mosquito and the microbe and the host too. And another thing important for as a living organism is that the lifespan of mosquitoes is important, and the time that the mosquitoes spent with interacting with the microbe is something important. On a bit anecdotal, but the fact that despite it's not a syringe, is flying, mosquitoes consider it as an active hunter. It will go forward towards its prey. And so that's one aspect of the behavior of the flying mosquitoes. The second aspect is the invasive species. I put indeed because the inherent capacities of the mosquito to fly is maybe not the best mean for invasion. And clearly, invasive species are species linked to human, and they take profit of human transport mean to invade countries or continent. So a bit of mathematics here for transmission epidemiology. I will go quick just to know that there's many factors in the transmission phenomena, which is the host preference. We have seen that. The vector competence, we will see that. I say the daily survival probability because the mosquito needs to have a sufficient long lifespan to accomplish the development of the microbes, and the extrinsic incubation, I'll repeat the word, the extrinsic incubation of the pathogenic agent. That means that the time that the pathogen will need inside the mosquito to develop. So another important thing is the difference between vertebrate and mosquitoes in terms of antiviral immune response. So in vertebrate, definitively the antibodies are a major part, secondary, but major part for adaptation to defense against microbe. The vertebrate interferent is also important in the primary response, and it could be predominant either in protection or sometimes badly in immunopathology. For the invertebrates, the immunity, the RNA interference is really very important in fostering of defense. And if you want to refer to that, I encourage you to read papers from Carla Salé at Pasteur Institute, who is really leading that field. We can consider there's no adaptive immunity in insects, but indeed microRNA could be a sort of adaptive immunity. And once again, I recall you papers from Carla Salé. So you see that between vertebrate and invertebrates, there are different evolutive constraints. Having said that, there are similarities. Both, have RNA interference, either by dicer or RIG1 receptor on helicase domain in the both sort of animals. There's sort of pattern recognition receptor too in both, on all the pathway like TOL, AMD and JAK-STAT pathway are common in the two sort of animals. So an example here to illustrate that is the interaction with West Nile virus on the left with vertebrates interference. So a cell is the virus is introducing itself in a cell sensed by PRR, and then a defense cascade with the production of interferon beta, which will warn the neighboring cells to express antiviral effectors through JAK-STAT signaling, etc. I won't go in detail for that, but just have a look on the left figure. So I have, that's the same figure because I want to underline the similarities, but clearly West Nile virus is entering a cell, a blue cell here. It's sensed by dicer2 on triggering RNAi cascade, but also the transmission of VAGO. VAGO is a molecule which will be transferred to a neighboring cell, so a bit like interferon beta in a vertebrate, and then VAGO will trigger again through the JAK-STAT pathway antiviral expression. So you see that's not the same molecule clearly, but that's very parallel pathways on IDs that both insects and vertebrates have found out in their DNA repertoire and dictionary. So once again, biological interaction. I told you a little bit time because, for example, the mosquito here as a cut is been the pathogen in blue is aspirated in the blood meal. Then it will develop in the midgut, mainly in the epithelium of the midgut, and then viruses will go out in the hemolymph of the mosquitoes, and finally will invade salivary glands where the delivery could be done towards another host. All that travel will take several days, depending on the pathogen. In case of malaria, for example, it could take 1 week or 10 days or even 15 days, something, according to the temperature. In case of virus, it could be very quick. For example, for chikungunya, it could make only 2 to 3 days. So it's very short, but usually for dengue, for example, we consider much more about 1 week or 10 days to have a complete travel through the mosquito organism. I put you to a picture of the brain, and you recognize the eyes, very lateral of the mosquitoes, and also the antenna first article, very dense. In red, there are location of Zika virus. You know, the mosquito whose head is pictured has been blood-fed with Zika virus, and Zika has spread towards the organism and inside the neural tissue of the mosquitoes. That's interesting that Zika, even in mosquitoes, have neurological tropism. That was an example.
John
On the
Bernard,
left,
John Bernard,
yes.
just for control, you have 15 minutes left.
Okay, so, okay, thanks very much. So, could we consider Amazonia as an exporter of virus? So, as I told you, it has been many imports in South America, yellow fever, we talk that, dengue virus, chikungunya, Zika virus. But is there any virus which could go global from Amazonia? That's the question I want to treat a little bit. So, here it's a sort of list of virus which have been found in Brazilian Amazon region associated with human disease. That's interesting because there are some arboviruses which are not described as triggering illness, but some others. So you see there's flavivirus, alphavirus, bunyavirus, phlebivirus, so large catalog of virus here. And so I propose you to think about two different candidates. The first one is the Mayaro virus. It has been described from Trinidad in the 50s. The first report in French Guiana is in 96. And when we look after on serological studies, indeed, it's very low in Cayenne on littoral, on the coastal line, but much more, about 25% in the remote village upstream of the big river, neighboring big river. So there have been urban cases during COVID on the coastline, so there was a bit of change in that. And also in a scientific station, there have been several cases which have been published recently with Loic. So clearly the pattern is fever, headache, rash, arthralgia, so nothing very close to chikungunya signs, clinical signs. Transmission is by hemagogus, whose is difficult to sample and to colonize. So here, an example of the serology I mentioned to you. So you have in red the coastline, and you see that for Mayaro, the serology is very low. Instead of chikungunya, it's about 20%. When in interior, high Oyapock, for example, Mayaro is about 20%, where here it's very low for chikungunya. So you have, some really interesting difference with an interesting paper to look after. Same paper, you see that in purple, you have the idea of timeline for circulation of Mayaro in purple, on chikungunya in orange. So you see that chikungunya clearly has been an outbreak in 2013, and in any geographical zone of Guyana, you have that, and you see that in high Oyapock or high Maroni, you have elevated transmission history of Mayaro, a bit continuous according to that, and that's related to the age serology you see in the B sector of the picture. So quickly, I will go forward. It's the animals which are found with antibodies to Mayaro, and you see that on the canopy you have the highest value. In the red sector, they are all monkeys, Alouatta, Sagenus saimiri, so squirrel monkeys or howler monkeys. They are the one which could carry Mayaro virus. The vector, Hemagogus gentinomys, is a very nice, another nice mosquitoes, purple one, trial breeder, day biter. So he's biting non-human primates and human. It's really hard to catch with dry eyes sometimes during the daytime, or human landing catch, being sure there's no Mayaro or yellow fever circulation for that. Experimental transmission of Mayaro, it has been shown with Egypti, some Anopheles, Aedes cupellaris, Aedes albopictus, so many things. But indeed, on the field, you see in Brazil, main strains of Mayaro are coming out from Hemagogus subspecies. So here to the clade, so there's different clades. There's 3 main clades, D, N, and L. In the arrow, red arrow are the clade of the French Guiana strain, which is in the Brazilian D clade. So another candidates are Bunyavirus. So there have been some cases of Oriboca Bunyavirus in the coastal areas. Mainly the sign as last dengue-like, so they are not evident, but all people are tourists or nature reserve guides and walkers. So meaning sleeping in the forest and having spent a night time in forest with different contamination sites around the same period between September and December 22. And entomological investigation we've done a bit later, 3 to 4 weeks later, show with light traps. We've seen, you know, the pie chart at the bottom left, that mainly that Culex portesi. You have an idea of Culex portesi. So Culex portesi, one Culex, that's an exception in Culex because he's easy to recognize with that you contrasted of clear and dark pattern in the thorax. But you see that mosquito is clearly the major part of that, and it's already an interesting candidate for as vector for this Bunyavirus like Oriboca. Tonat is an alpha virus of the Venezuelan encephalitis, equine encephalitis complex. So another Bunyavirus is Oropuche. So there have been an outbreak in 2020. During COVID times, there was no illegal gold mining control at that time because of COVID, no army camp in that region where usually there was. And there was been several reported cases by the local health center, confirmed by the virology lab in Pasteur Institute, who has just designed the right tool to make the diagnostic, the molecular diagnostic for Oropuche. You have here the chart for all the clinical cases with the investigation with hospital with a total of 37 compatible cases. So that means not so much, but indeed when you know that the village at that time has about 70 to 80 inhabitants, that made a huge attack rate. So which was the vector? You know, classically, the literature said that Coquilletidae venezuelensis and Aedes serratus is the vector in the sylvatic cycle, and Culicoides paraensis and maybe Culex kinkefaciatus in the urban cycle. So just a word of Culicoides. Culicoides are really small, tiny, biting midges, which are really hard to trap, especially paraensis, because he's a daytime biter again, and he's strictly, he's very much attracted by human. Well, that's one thing. So which control mean for vector we could find? So we make a field investigation with many traps, you know, BG Sentinel, CDC, light trap, mosquito traps, and larval search. And indeed, we find merely almost Culex kinkefaciatus on one Culicoides paraensis, which was a bit frustrating, but as you know, as I told you, it's very hard to trap and to capture.
5
All
minutes
was negative.
left. 5 minutes.
Okay, thanks. So excreta count and saliva count was negative, and we tested Culex kinkefaciatus, and they were negative also. So, but indeed, in 22, Oropuche, there was a mention of nuisance by numerous midges in Sahel, and we trap a lot in the villages, more in the center than the outskirts, but in all sites we find, and we find it on ourselves in a human landing catch, because we know there was no more Oropuche circulation there. So indeed, the vector paraensis is present in soil in July, so corresponding to the same time as the outbreak in 2 years before, and that's very compatible with the known vector role of Oropuche. So just to name the 5th episode of Oropuche is in 2024. You see here in Brazil in blue, that's a big outbreak there, but also in Cuba, which was new, about more than 1,000 cases in Cuba, also in Peru at same times, also in Bolivia. So you see, in that case, clearly the virus has been exported because Cuba was the very first time that the virus was described. Here you have a phylogenetic analysis, a tree, where at the bottom you see the Cuba strains from 2024, and just above the strain of French Guiana in 2020, and you see it's clearly related, all with Brazil in the 23 and 22. All that is a cluster, clearly, which has been shared between Cuba, Brazil, French Guiana, and I would say probably Peru and Bolivia, but we don't have a recent strain for that. Just to mention that the orthobunia virus, like Oropuche, have 3 segments. They can exchange segment and reassort between different viruses, and that makes the things a bit more complex that we use. So clearly, a few words, sorry, Paula, from the AMAZ project. So it addresses sampling in animal vectors on human, and then virus discovery and virus evidence by NGS, by high throughput technique with fluidic, microfluidic PCR, and vector competence in BSL-3 tests. All that data will allow the building of risk model and delivery to the stakeholders and society. So I won't detail here a photo of sites around Cayenne where there's a mix of a mosaic of forest on villages, on urban areas. So it's very interesting for the interface. And just to show you one result is that in the light green and the dark green, you see roughly the distribution is the same according to the different sites. And that means that either in the forest or at the edge of the forest, the composition of mosquito fauna is roughly, on the abundance is roughly the same, where outside of the forest, clearly in the villages or even in savanna, you have a big decrease of that. So I told you about the NGS, just to mention that the Pasteur Institute in Paris is very much involved with Nolwenn Dely's team, Carla Saleh's team, on the ANSES with Sarah Moutaillet's team for digging out a virus from that, and also for Dominique Rousset team in Cayenne, who is helping us to find some virus in the mosquitoes. Just, I forget to mention, there was different sites, Guyana, Guadeloupe, New Caledonia, and Cameroon, too, where all the same methodology are applied we will want to share results and to find common factor for spread of virus outside of the forest. So just one word, interesting work with the vector competence of Coquillettidia venezuelensis. I told you about that vector. So we try, as it is known well in the literature as vector for Oropuche, we try to infect it, and we indeed didn't find it able to transmit Oropuche in the saliva. So that's very first result for that, interesting for maybe digging out from the list of potential vector for Oropuche that Coquillettidia venezuelensis. Yes, I won't go towards that. So conclusion, taking home message, as probably you understand, that's not a flying syringe on their specific interaction. So by nature, that's really a One Health problem for that virus, arbovirus in Amazonia. So Amazonia could be an exporter, clearly. Oropuche virus has been an example. I think we have to keep in in the loop for other Bunia virus, local Bunia virus, just in case of. The perspective are, so the Amazons next season, it will go, I hope we will find, give you more data on, clearly I put it goes virus, not very funny, but I mean that just because the virus discovery will go really at a deeper, on a higher level now. And just another project I won't have time to describe, which is about mangrove, the Magellan project with Medicole in photo here on left, addressing mosquitoes, but also culicoides, and that's something with very new things. So please look after for papers soon from Amandine Guides, from Stanislas Talaga, Medicole. Arthur Delille is a human science scientist, Alizé Lagrave and Dominique Rousset from the Virus Lab in Pasteur Institute. And I think I have finished for my presentation. Thanks very much for attention, and thanks for partners here. My beautiful team on the top left, and all the partner and founders we have in the lab. Thanks very much.
Thank you. Thank you very much, Jean-Bernard, for this very insightful presentation highlighting the importance and the impact of your research on mosquitoes. And I'm thrilled to see that we have a lot of questions in the chat. So I would invite those who wrote the questions to read them if you have the possibility, because I know some of them maybe have difficulties with the connection, with the internet access. So if you are able to turn on the camera and read the questions, that will be really nice. Otherwise, I can read the question for you. So we have one question from Ubimo Paggy Patrick. Patrick, are you able to turn on the camera and ask your question, or do you want us to do it for you? Well, I think I'll do it. So this question is in French. So, est-ce que l'étude que vous menez dans l'Amazonie française ne peut pas être menée sous votre financement, j'essaie de financement, bien sûr, dans des milieux ayant une écologie similaire, comme en République démocratique du Congo, par exemple?
Alors, je réponds en français ou en anglais?
English. We will keep it in English.
Okay, we keep in English. So, yeah, definitively, Patrick, we have that idea on in the Amazon project. I went very quickly. I'm very sorry, because that's a huge project, and indeed the Cameroon is involved in that project. So there's a part in the south of Cameroon with a mix of forest, villages on palm trees plantation, where the same sort of sampling for mosquitoes, and then after for virus discovery is done exactly, and we will try to share our thing. The leader in that is Christophe Poppy from IRD with local partners in Cameroon. So why not in Congo? We don't have the funding is a bit strict here, so we can extend the region right now. But the idea of having other region with sort of studies like that, I think that's definitively something interesting. And you should look after in the PRESOD initiative towards that.
Thank you, Jean-Bernard. We have also another question from Eric Ledroux. Are human pathogens associated to Coquilletidia?
So definitively there have been some isolation from virus for Coquilletidia. So I mentioned Bunia virus on Oriboca, for example. There's some other sort of Bunia virus which is relatively complex in terms of systematic, and I won't go further for that, but there has been several strains of Bunia virus, not only strain, but species, I can say, which have been associated with Coquilletidia. Also, I think that tonnads, the one I mentioned about the Venezuelan equine encephalitis complex, has been two. The question is that where viruses are found within mosquitoes, and clearly the vector competence we have done is logical with the fact of finding, you know, Oropuche in Coquilledilla venezuelensis, but indeed it won't go up to the end of the transmission chain with the saliva. So it is associated. It is associated with carriage, but in my mind, our recent work show it is not associated with transmission. Is my response clear for Oropuche? But I mean, we have to test for other Oriboca, for example, Bunyavirus, and even some other tonat, for example, virus, etc.
Thank you, Jean-Bernard. We have another question from Demeki Daka Ketere. Would you like to read the question? Demeke? So he's asking, he's connecting from Ethiopia, and his question is,
Yeah, I
human
see that
landing.
about the,
Okay,
yeah.
okay. So
yeah. About the
no,
ethical...
there he is. So please turn on the camera and go ahead. Thank you.
Okay.
Thank you very much. I'm Demeke from Ethiopia, and I am PhD student at Jimma University. Especially my background is what about medical parasitology, and now I'm PhD student in tropical and infectious diseases. My question is related to the ethical issue during the mosquito collection. You know, we all know human landing catch is one of the best mosquito collection technique. But related to that, there is a serious ethical concern. We are exposing our participants to a lot of vector-borne diseases. So due to that reason, even institutional ethical review board rejects a lot of students' proposals. So how we manage this issue properly? I think this is my question.
Yeah, thanks very much. I totally agree with that concern. That's a major one. When I work on malaria too in West Africa, WHO definitively try to reject that technique for malaria vectors because there was some other means. In the case I present to you, the yield for capture with trap for example, Culicoides parensis is very low, except if there's huge population, when in that case they sort of spill from the huge population towards some in the traps. So that's a major concern. The concern for us is first to be sure there's no circulation of the virus at the time and the location where you site for human landing catch, and you have to be really solid and robust about that affirmation. The second possibility is to try to protect with vaccine. And the third one is to protect with a mechanical mean, like, for example, a bed net. You see, you can have I'm sorry to employ that term, but human bait, a person who is sitting on a chair under a bed net, and then another one which is very well protected and which can aspirate mosquito flying around the bed net for that. So that's the possibility to have that. And I know that in Brazil, they use that types of either double bed net or the sort of bed net plus aspirator around the protected bait. Excuse me for that term. Is it okay?
Thank you. Yeah. Thank you very much. I have another question for the training prepared groups, or which means postgraduate members. I think a lot of students from Africa are working a good research, but there is a lot of budgets as well as a lot of problems are there. Even if there is a lot of skills based on a lack of trainings. So this is the webinars and the related things and the seminars are very important to us. So my question to them is related to this problem is either any special training, which means face-to-face, like the molecular techniques, are there from your side? This is a question for all of the African students. Thank you very much.
Well, there's some training. For example, the Institut Pasteur in Paris is offering training in medical entomology. I think it's a 4 weeks training on site in Paris, so you have to go there but it's really open to any countries, and so I think it is in English. I have to check about that, but I think so. Otherwise, there could be, local course or training session. I know, for example, Dakar is very expert in medical entomology on arbovirus, so there may be something here. I don't know. I'm not updated on these offers, but you could find that for the molecular biology, we make a collaboration. For virus, it's much easier to do, make the other work for you. It's the best way to have results. And so, no, I mean, I'm joking, but if it's very complex, NGS or something like that, we do a bit here on locally, but where the work is going upstream with upper scale, we have to collaborate with other teams which are dimensioned for that, where we are not. So on the other things for cytochrome oxidase sequencing, that's basic molecular biology. That's not really specific for mosquito, but I don't know if I answered your question. I'm sorry, I'm not really precise on that.
Well, we have another question from Julie. Julie, could you please turn on the camera and ask your questions directly?
Hello, Jean-Baptiste. In English, so I think my question is almost answering because it was about the identification of mosquitoes. Mostly, I think you have a lot when you catch in field. And so how did you do to identify the mosquito? Do you, mostly for AMAZ program, do you do
in French, if you want, it's okay too. We just want you to feel as comfortable as possible.
Maybe a first identification with a loop, and after you make a bio identification in your lab for mosquitoes.
Yeah, that's a big question of work process. Because in that case, for example, in AMAZ, I didn't mention, but one day, one unique trap catch 3,000 mosquitoes.
Yeah.
And in that case, the technician in the lab are really bad words toward me, because that's a hard work. That's really hard work. So indeed, I didn't precise that, but we use cages instead of, a net for trap, and then surrounded by plastic bag to keep a bit of humidity inside for the best survival for the mosquito, for them to be fresh. And then we bring them back in the lab, and then in the lab we aspirate some, we put them in the fridge and to cool them down, and then we identify them. And that's a huge change, that sort of trap with 3,000 mosquitoes that takes 2 full days to 3 experimented technicians to sort out everything. So that's a very hard one. So usually there's a first line with a quick sorting out for mosquito or other insect or whatever. Then after by genus, and then after for the Culex genus, which is a more complex, we go to morpho species first. And after, in case we want to go further, we have to genotype, so make pool, and so very complex. So currently we make pools for group of Melanoconio subgroup. Sometimes we have specipes group or something like that. Okay, or so we can do that. But at the point we have chosen to say we will make virus research and then go back to the mix, because we are obliged to mix everything, to go metagenomics for mosquitoes. It doesn't work perfectly, but that's the how to compromise with that.
Okay, so you can make a morphological identification with genitalia or something else.
Genitalia is only.
Yeah.
Genitalia is only for males, so males are not transmitters, so they can be infected vertically or sexually, but they don't bite humans. So we are not really interested, except for defining the spectrum of species which are present at a given site at given time. You see?
Okay.
So that's a sort of balance between time devoted to that and then the result at the end. And that's always a difficult choice. I agree.
Thank you.
Just one option, which is very big time, is to keep some legs for each mosquitoes in a tube, and then you pull them, you search for a virus. If it's positive, you go back to the legs and you say, well, there have been that species present. That's huge work. And you need big place in freezers. That's so stupid as a limit, but your freezer is on boxes, on tubes, and whatever. That's a very real life limitation,
Well, thank you all. It is time for us to end the session. I was just to let you know, Jean-Bernard, there is a kind of invitation for me. I see it that someone is saying in French that nous avons plusieurs cadres qui travaillent sur ces différents arbovirus. Un partenariat peut-être très bénéfique avec l'Institut de recherche en biologie appliquée de Guinée-Kindia. Donc là, je pense que c'est peut-être une discussion qui pourra se poursuivre par email ou sous d'autres formats. Mais je ne voulais pas laisser les commentaires sans le présenter. Nous vous remercions. Thank you all for joining us and for attending this monthly session. Just some reminders for the next webinar series from the graduate school. So remember that the next Wednesday, March 25th, the social science webinar of the graduate school One Health and emerging infectious diseases will present Ilona Lowy, a historian of science, and she will talk about viruses, mosquitoes, and reproductive injustice, Zika in Brazil. That's for next Wednesday. And of course, as I mentioned before, we will have on April 22nd, Professor Guillaume Odana. He will talk about an ethnomedicinal approach to major Amazonian diseases. On May 18, Professor Antoine Adeny, histoplasmosis in people living with HIV in Latin America, and on June the 24th, Professor Paul Letournier, leptospirosis in French Amazonia. Thank you all for attending this session, and we stay tuned for more exciting webinar series.